Hello, my name is Lukasz Spiewla. Last summer, I researched the nocturnal stomatal conductance of Carex Oligosperma through Michigan Tech's SURF program in partnership with the U.S. Forest Service Northern Research Station. A little bit of background information to my project. It's well known that peatland ecosystems act as highly effective carbon sinks. This is due primarily to the low oxidation and decomposition of organic matter because high water tables limit aerobic transpiration. To counteract high water stress, Carex Oligosperma have adapted aerenchyma. These are modified roots which create a direct pathway for gas exchange from the stomata to the roots. There's evidence that rhizosphere microbes capitalize from the steady supply of oxygen to perform aerobic metabolism around aerenchyma. This effect results in decreased methane production and allows for greater processing of organic matter, changing the ability of peatlands to act as carbon sinks. Based on the water conditions created by peatlands, I hypothesized that the stomata of Carex Oligosperma would remain open through the night regardless of water table depth and surface peat moisture. The conditions of the U.S. Forest Service tree peat experiment created a good experimental model with established populations of Carex Oligosperma. The primary method for collecting data was through direct comparison between night and day measurements from full population samples. Live sedges in groups of 3 stalks were measured by diameter and marked in the bins prior to measurements. Once secured and stabilized in the lichor, conductance measurements were logged against varying conditions of humidity, temperature gradient, and vapor pressure deficit. To supplement the data collected from the midday and midnight measurements, light response curves were also measured alongside midday measurements. Photosynthetically active radiation, or PAR, was measured on a stock bundle with each daylight measurement at 9 different levels of PAR, ranging from 0 to 1500. Light response curve additionally estimated the effects of PAR under 50, or low PAR, on active plants in daylight. In order to further investigate the minimal stomatal conductance of Carex Oligosperma, abscisic acid was used to induce stomatal closure in plants in day and night. By using abscisic acid solution, a minimal stomatal conductance value was able to be determined for Carex Oligosperma. 3 stalks were measured, cut, and placed into 2 vials of either DI water or ABA solution for each bin sampled. Stalks placed in ABA solution quickly saw changes to conductance within an hour of being placed in solution. It's worth noting that ABA can determine a leaf's minimal stomatal conductance, but cuticular conductance, as you can see in figure 4, requires further measures to isolate and should be considered separately. Based on our findings, it's apparent that stomatal conductance was very sensitive to low light, seen in figure 3. Furthermore, we can conclude that the stomata of Carex Oligosperma are neither fully open nor fully closed at night. Carex Oligosperma also shows a higher than average stomatal openness than other species at nighttime, as you'll see in figure 4 by the red marks, and does not exhibit major differences in stomatal regulation based on water table depth. Finally, I'd like to thank a few important contributors to this project, including Drs. Molly Cavallari and Hilda Smith. I'm especially thankful to Drs. Eric Lilaskov and Colin Tucker for advising me through this project. I'd also like to thank the U.S. Forest Service for providing a facility for this project, and Dean Andrew Storer for funding this project.